the last section, we have learned something about the basic knowledge of DNA and the genetic markers. Congratulations, you have made the first step to a DNA analysis. In daily work, the forensic DNA analysis consists of a series of complicated procedures. Here is a basic outline of the procedure for forensic DNA analysis. So in this chapter, we will talk about some basic techniques for DNA analysis, including the DNA extraction, the DNA quantitation, the DNA amplification, and the electrophoresis. The source of DNA can be any biological sample containing nucleated cells, such as the white cell in the blood, the saliva, the mouth epithelial cells, the semen, hair, and the bones and the tissues. A biological sample obtained from a chromosome in the form of a blood or a semen stain, or a liquid blood sample from a suspect or paternity testing case contains a number of substances besides DNA. DNA molecule must be separated from other cellular material before they can be examined. Cellular proteins that package and protect the DNA in the environment of cells can inhibit the ability to analyze the DNA. Therefore, DNA extraction methods have been developed to separate proteins and other cellular materials from the DNA molecule. There are three primary techniques for DNA extraction used in forensic DNA laboratory: the organic extraction, the Chilex 100 extraction, and FTA paper. The exact extraction or DNA isolation procedure varies depending on the types of biological evidence being examined. For example, the whole blood must be treated differently from a blood stain or a bone fragment. The organic extraction, sometimes referred as the phenylchloroform extraction, has been in use for the longest period of time and may be used for the situation where either RFLP or PCR typing is performed. High molecular weight DNA, which is essential for the RFLP method, may be obtained most effectively with the organic extraction. The Chilex 100 method of DNA extraction is more rapid than the organic extraction method. In addition, Chilex 100 extraction involves fewer steps and the fewer opportunity for sample-to-sample contamination. However, it produces single-strand DNA as a result for the extraction process, and therefore is only useful for PCR-based testing procedure. Chilex 100 extraction is an advantage for PCR-based typing method because it removes the inhibitors of PCR and use only a single tube for the DNA extraction, which reduces the potential for laboratory-induced contamination. Another approach to DNA extraction involves the FTA paper. The FTA paper involves adding a spot of blood to the paper and allowing the stain to dry. The advantages of FTA paper are that consistent results may be obtained without quantification, and the procedure may be automated on a robotic workstation. Determining the amount of DNA in a sample is essential for the polymerase chain reaction, that is PCR, based on DNA testing. Because PCR-based DNA testing is very sensitive, a narrow concentration range is required for the application to be successful. If too much DNA template is used in the PCR, the resulting artifacts may interfere with the data analysis and the interpretation. On the other hand, very low amount of DNA template may result in a partial DNA profile or the failure to attain a profile. Also, forensic evidence samples are often mixtures that may include DNA from non-human resources. Thus, this is very necessary to use a human-specific DNA quantitation method to selectively determine the amount of human DNA present. Early method for DNA quantitation typically involves either absorptance at a wavelength of 260 nanometer or a fluorescence after the stain to yield a gel with ethylene bromide. Unfortunately, these approaches are not very sensitive. In addition, absorptance measurements are not specific for DNA, and contaminating proteins or phenol left over from the extraction procedure can give false high signal. To overcome this problem, several methods have been developed for DNA quantification purpose. These include the slot bot procedure and the fluorescent-based microtiter plate assay, as well as so-called the real-time or quantitative PCR process. Forensic DNA typing laboratories have greatly benefited from the discovery of a technique known as the polymerase chain reaction, that is PCR. PCR is an extremely powerful method that allows selective amplification in vitro of specific target DNA sequence from large heterogeneous sources such as the genomic DNA. The PCR is an enzymatic process in which a specific region of DNA is replicated over and over again to yield many copies of particular sequence. Here is the PCR mixture, or we usually call it the PCR system. In order to be able to select a particular target from the mixture of sequence, specific sequence information must be available. This information is used to synthesize short oligonucleotide, we call it the primers, that initially bind to the complementary sequence. The reaction mixture also contains the DNA building blocks that the 4-deoxynucleotide triphosphates, the DATP, DCTP, DGTP, and the DTTP. DNA polymerase produce a complementary strand by connecting the deoxynucleotide triphosphates according to the sequence information given by the template strand. The buffer containing magnesium ion and the DNA polymerase used is the one chosen for its heat stability, so that it survives the heating steps of the cycle. Each PCR consists of 3 steps. At the start of the progress, all reaction partners must be single-stranded, which is achieved by the heat denature. We call it melting. Subsequently, annealing primers molecule to the template DNA. It happens at the particular temperature, which is decided by the structure of the primer and the template. And the third step, the deoxynucleotide triphosphates continue to add, elongating the strand at 72 centigrade to form a new strand. The newly formed double-strand DNA molecular are heat denatured again and can themselves serve as a template in a new cycle of DNA synthesis. During each step, the temperature and the duration of the reaction must be carefully controlled, which is most easily achieved by the using of automatic devices. One cycle will normally require only a few minutes, depending on the equipment used. The final result is that only the DNA sequence located between the 2 primers is eventually synthesized. Theoretically, after 30 cycles, 1 billion copies of target region on the DNA template have been generated. This PCR product sometimes refers as an applicant as then in sufficient quantity and quality, that is, can be easily measured by a variety of techniques. The method is extremely robust, that it works with very small amount of DNA. even if the DNA is badly degraded. For example, a single cell and biological samples that were dried, mummified, buried, or stored in the paraffin box. PCR has been simplified in recent years by the reagent kit that allow a forensic DNA laboratory to simply add the DNA template to a pre-made PCR mixture. The kits are typically prepared so that a user adds the liquid of the kit solution to a particular amount of genomic DNA. The best results are obtained if the DNA template is added in an amount that corresponding to the concentration range designed for the kit. In the kit, controls are usually used to monitor the effectiveness of the experimental condition and the technique. A negative control is an entire PCR reaction mixture without any DNA template. The negative control usually contains water or buffer of the same volume as the DNA template, and is useful to assess whether or not any of the PCR components have been contaminated by the DNA. An extraction blank is also useful to verify that the reagent used for DNA extraction are clean of any other DNA template. A positive control is a stranded DNA template of known sequence with good quality. This is a valuable indicator of whether or not any of the PCR components have failed or were not added during the reaction setup phase. It also ensures the reaction components and the terminal cycling parameters are working for the application. Furthermore, PCR reactions have been advantaged that they can be performed in multiplex fashion, which means that different forward and backward primer pairs are mixed to allow different target sequences to be amplified simultaneously. The simultaneous amplification of two or more regions of the DNA is commonly known as the multiplexing or multiplex PCR. Multiplex PCR is more of a challenge than the single reaction because so many primer annealing events must occur simultaneously without interfering with each other. Extensive optimization is normally required to obtain a good balance between the various losses being amplified. To this end, primers are designed in such a way they are locus-specific. Even when used in identical reaction conditions, the result of the multiplex PCR is a mixture of the product which are nevertheless distinguishable in subsequent analysis by electrophoresis size separation. Also, the PCR can be improved by the fluorescent detection method. Advantages of the method include a higher sensitivity and a broad dynamic range than the comparable colorimetric detection method. Furthermore, they have the capacity for simultaneous multi-parameter analysis of complex samples such as the multiplex PCR product with different fluorescent labels. The range of the common fluorescent dye used in DNA labeling is between the 400 to 600 nanometers. Fluorescent dye labeling can be incorporated to a DNA fragment. Use a 5-end labeled primer. The dye-labeled primer method is usually used for STR profiling, in which only one primer is labeled. Therefore, only one strand is visible. Additionally, dye-labeled primers allow multiplex PCR amplification in the same tube. It is necessary to separate wire size of DNA fragment. This can be achieved by the electrophoresis technique. Fragments are separated based on the migration of charged macromolecule in an electric field. DNA is a negative charged molecule with the phosphate group of DNA nucleotide carrying negative charges. DNA migrate from the negative electrode towards the positive electrode in the electric field during the electrophoresis. The electrical potential is the force responsible for the moving of charged molecule during the electrophoresis. Thus, proper ionic strength of a buffer is necessary to achieve efficient conduction. The mobility of a macromolecule is primarily determined by the charge to mass ratio and their shape. However, because the first phase group of every DNA nucleotide carries a negative charge, the charge to mass ratio of DNA is same even the length of DNA fragment varies. The forensic DNA testing usually analyzes linear DNA, and the shape of linear DNA are similar. Therefore, the separation of different sized DNA fragment is based more on the effects of a molecular sieve mechanism provided by the supporting matrix. It is obviously easier for smaller molecule to migrate than the larger molecule. That's why they migrate faster through a matrix. Hence, the mobility increases as the size decreases. Conversely, larger DNA molecules migrate much more slower because they experience more friction as they travel through the night of pore of a gel. Therefore, the separation of DNA with different sizes can be accomplished. Capillary electrophoresis is a newer method. Separation in capillaries is performed at high voltage. One essential component is the capillary. This is a thin hollow tube made of fused silica with the translucent detection window to detect the signal from the labeled DNA fragment. During the electrophoresis, the capillary is connected to the buffer sessile that are connected to the electrode. The injection of the samples is performed by an autosampler whose injection are based on the charge of molecule. Only small quantity of the samples are required for each injection, and any remaining samples can be reused. Capillary electrophoresis instruments are equipped with the detection system utilizing laser excitation source that excite the labeled DNA fragment. They also include the fluorescent detector that record the signal sent by the fragment. The argon ion gas laser is frequently used in the applications such as the fluorescent label STR analysis because typical fluorescent dye match the excitation properties of the argon laser. Optical filters are usually to detect the light emitted only at a particular wavelength. Signal from the multiple fluor in the same sample can be recorded separately. Use the optical filters and the mathematical matrix. The function of the matrix is to subtract the color overlaps of the layers of fluorescent dye. A fluorescent detector is a photosensitive device that measures the light intensity emitted from a fluorifer with the photomultipolar tube or charge-coupled devices. The signal from the fluorifer is collected and converted to the electronic signal. Thanks to the above technique, by using them, we have enlarged DNA copies in virtual for billions of times and easily convert the invisible information that we collected in the chromatin to visible signal and picture. Next section, we will talk about how to interpret this signal and transfer them to the data we can use. Thanks for watching.